Welcome and good afternoon, everyone. I'm Patricia Borger, Vice Chancellor of Development and Alumni Relations at the University of Wisconsin-Milwaukee. I'm so pleased that you could join us this afternoon for this very special event. This is our 8th annual Chancellor's Innovation Award ceremony, and it's an event that always shines a spotlight on some truly visionary individual who's made a positive difference in our lives. Today, we're joined by leaders of business, education, government, public health, and healthcare. And I know that many of our faculty, staff, and students from UWM are here as well. So welcome to you all. I'd like to give a special thanks to the Argosy Foundation for making this event possible, especially Chris Abley and Emily Van Dunk. And a special welcome to Cheryl Sikora from the Argosy Foundation, who's joining us virtually today. Usually we do this event at a luncheon. However, in light of the pandemic this year, here we are online. We know that the benefit of our being online, of course, is that we can welcome individuals from across the country, including our honored guest today. Now I invite our Chancellor to join us. Please welcome Chancellor Mark Money.
Thank you, Pat. Thanks for your great leadership and the work in establishing the Chancellor's Innovation Award and really being able to recognize such remarkable individuals like Dr. Corbett, who I'm pleased to introduce today. This is a special event. It's about success. It's about individuals who are change makers in our lives and in so many different ways. You know, the heart of this is about innovation. And just as a quick comment, we had Vice President Kamala Harris on our campus last week, where we were able to showcase, as part of her and President Biden's initiative to bring $180 billion in research and development money through federal funding to campuses like ours. So we were able to showcase how we train students to engage in creative thinking, how we have incredible faculty and scholars doing work that changes lives every day. But I'd like to talk a little bit about this award in the context of why we think it's such a wonderful honor for Dr. Corbett today. Previous Innovation Award recipients include Microsoft CEO Satya Nadella, who happens to be an alumni of UW-Milwaukee, founder and CEO of Bird Health, and former CEO and president of Safeway for 14 years, Steve Bird, Major League Baseball Commissioner Bud Selig, retired Bath and Body Works President and CEO Beth Pritchard, Academy Award-winning writer and director John Ridley IV, WEC, Wisconsin Energy's Energy Group Executive Chairman Gail Klappa, and Fiserv President and CEO Jeff Yabuki. Now, half of those individuals who I just listed are UWM alumni, and we're very proud of them. But the others all have ties to UW-Milwaukee through either business, community involvement, philanthropy, or other ways. And you might be asking, well, so how does Dr. Corbett have a tie to Milwaukee or Wisconsin? Great question. Here's the answer. Have you been immunized? Are you thinking about getting a vaccination? If so, you are somebody who's a recipient and a beneficiary of the great work that Dr. Corbett has done. You're now able to go out to eat again. Maybe you're together with relatives, family, friends. You're able to get back to normal. This is the woman who has really helped us get our lives back. Dr. Kizmikia Corbett she's an immunologist at the National Institutes of Health. She's one of those scientists who helped to develop the COVID vaccine. In fact, her work was instrumental in creating the Moderna vaccine. If you got that vaccine, you can thank Kizmekia, our guest today. She and Dr. Barney Graham led a team of scientists to develop the vaccine with 94 to 95% efficacy. That's incredible, and thank you, Dr. Corbett, for that work. Dr. Anthony Fauci called her a rising star in his tribute piece that he wrote when Time magazine named Dr. Corbett to its 2021 Time 100 next list of emerging leaders. This is a wow. Dr. Corbett got her bachelor's degree at the University of Maryland, Baltimore County, and she went on to earn her PhD in microbiology and immunology at the University of North Carolina at Chapel Hill in 2014. In fact, I just learned that Dr. Corbett is going to be receiving also an honorary doctorate from the University of North Carolina next week. Congratulations, Dr. Corbett. Since then, Dr. Corbett has been a research fellow at the National Institutes of Health, focusing on respiratory virus vaccine design. Her work has been profoundly important to many of us on a personal level, and it's been critically important on a national and international level as well. But there's more to her story. In fact, it was just yesterday announced that Dr. Corbett will be joining the faculty at Harvard. And let me just share with you a few excerpts in terms of recent releases. This is the ultimate in late-breaking news, and I'm just delighted. So from the most recent update, Harvard T.H. Chan School of Public Health today, this was yesterday, announced that Kizmikia S. Corbett will join the school as a named professor in the Department of Immunology and Infectious Diseases. She'll hold an appointment at the Harvard Radcliffe Institute as the Schuster Assistant Professor. Corbett comes to Harvard Chan following more than 6 years as a research fellow, and you know some of the things that I've always talked about. There's two important quotes that I'd like to share from her work. She said, "Vaccines are the great equalizer when it comes to addressing health disparities, especially around infectious diseases. Harvard Chan School is at the forefront of advancing health equity, and I'm excited to join such distinguished colleagues in my pursuit of viral immunology research and vaccine development." Comment, I have to tell you, as a research one, and especially as an Access University, UW-Milwaukee wholly endorses and finds incredible alignment with Dr. Corbett's core value on this front. This is exactly why we've established the Vaccine Center and why on UW system campuses, you have over 50% of the testing that's been done in the state of Wisconsin. And as you know, equity and inclusion is a critical part, not just of how we operate on our campus, but truly in terms of how we engage our community, trying to reduce disparity and close achievement gaps. A quote from one of the institute's deans at Harvard said, "Kizzmekia is an inspiring scientist who, though still early in her career, has already had incredible impact. Her work is helping to reshape our world and is saving lives on a vast scale." This is pretty amazing type of commentary, and we're just so excited to see the continuing contributions, Dr. Corbett. She's also among a group of 60 Black members of the National Academy of Medicine who spend their free time, and I can't imagine there's much free time, talking to communities of color about science and vaccine hesitancy. When you do have that time, I find it so neat how much you care about others and informing individuals about their health choices. But it wasn't just this year, and that's what's really important to me. Dr. Corbett has been talking to communities about science since she was 20 years old. She has connected with groups from Girl Scouts to churches, reaching out and relating to people in order to help them. She's keenly aware of health disparities for people of color, and this awareness has motivated so much of her work, producing vaccines that help everyone equally. We're so much in alignment with our mission about access and education for individuals from all backgrounds. And we know that it's education that is the harbinger, it's the predictor of so many of life's achievements and life opportunities, from home ownership, social mobility, but particularly health outcomes. And so that's why we find such great alignment. With those comments, I'd now like to welcome Dr. Kizmikia Corbett and ask her to speak a bit about what has driven her innovative work. Dr. Corbett.
Thanks so much for that kind introduction, and thanks to everyone who's tuning in, albeit with a slight schedule change. I am absolutely delighted to be with you today, albeit virtually, but I will say that I'm very happy to take you up on lunch after we start back traveling and after you all get back to the point of where you're inviting people back to your campus. I have never been to Milwaukee before, so why not? I am Kizmikia Corbett, and I guess I can say, as was just mentioned, I am my screen is disabled, but I am a rising assistant professor at the Harvard Chan School of Public Health, where I will be studying essentially the same things that I'm studying now, except broadening my horizons just a little bit to more viruses outside of the coronaviruses. And the work that I'm going to talk about today is the work that I've done at the National Institutes of Health and the Vaccine Research Center, which is this building in the background of this slide. It is building number 40 at the National Institutes of Health's campus in Bethesda, Maryland. And I'm going to just give a snippet of what was and is actually still the race to the vaccine that was fueled by pandemic preparedness. And because this talk is supposed to encompass what has prepared me, I decided to add a little bit of career preparedness as well. So let's start with the career part. Even though I'm only 35, I've actually been in a laboratory setting for about 19 years now. Almost all of my life, at least since high school, where I took part in a summer program called Project Seed, which allowed for me to do research every single summer at a Research One institution, like the institution that you are at, where I was awed by the scientific process, by the day-to-day life of a scientist, and being able to work on a therapeutic and a novel one actually at that time. It just seemed like this was something that would be so good to do with the rest of my life. And so from that moment on, I did a stepwise career trajectory towards the goal of where we are or where I am right now. So next on that timeline, what I did was I went off to the University of Maryland, Baltimore County, where I was a Meyerhoff Scholar. And I continued to do work actually at the National Institutes of Health during that time as well, both in the summers and then also on our 1 month winter break, I would go to the NIH and do research. And that's where I fell in love with government research and the process of government research, where there is so many resources, so many talented scientists in one place right next door to Washington, D.C., that really drive forward a lot of innovation in this country. And so I said, I want to come back to this place, but obviously before I could do that, I needed to go off and get my PhD. And so I went back to home to North Carolina at the University of North Carolina at Chapel Hill. And I got my PhD in microbiology and immunology. And then I became a research fellow and now a scientific lead for the coronavirus team at the Vaccine Research Center, which I will talk about my work. But one of the things that I wanted to make clear about this slide, especially, talking to academia and at a university that clearly puts forth a lot of efforts into honoring innovation and also honoring the career trajectory of their students. I wanted to make sure that on this slide, it was very clear that at each step of the way, I was backed by a program or a university specific program or a scholarship that really helped to fuel my career into the direction of which it was now, even down to the fact of the Office of the Director at the National Institutes of Health partially funding my fellowship in the beginning of my postdoc. So I am at the Vaccine Research Center, which was founded in August of 2000, and the entire purpose of that building was to develop a vaccine for HIV/AIDS. And then obviously we don't have a vaccine for HIV/AIDS, although the field has completely transformed over the last 21 years. The main thing that comes out of what we've learned about making a vaccine for HIV/AIDS is that the Vaccine Research Center now has a full and vast portfolio of vaccine and therapeutic products for multiple different viruses, all of which are listed here. And you can tell from, for example, Ebola or Zika, or even the coronaviruses, that many of them are in this what's called pandemic preparedness. So they are viruses that have reemerged or are continuing to emerge all over the world. The one thing that struck me very about the Vaccine Research Center is this really unique ability to go from a protein like you see on your screen and like we did with the coronavirus vaccine development all the way through to advanced partnership. And when I took on this coronavirus project, honestly, the truth of the matter is I never thought that I would get to the point of the advanced partnership. But I did always know that because the virus was so understudied, because the literature was essentially a blank slate, that I did have this kind of niche, this niche area for which I could create and be, as it said, innovative in that space. And so I did an interview recently for a government leadership award, and I kind of explained that choice.
6 years you've been involved in this kind of research. What made you pick this field?
The exact opposite of what's happening right now. No,
truly. When I was interviewing for research fellowships and I talked to Dr. Graham during my interview with him, one of the things that was very important to me is that I work on a project that I would be able to create my own niche in, because to move upward into principal investigator position in science, obviously you have to have a niche. And the point is to become the world's expert in that thing. And if someone needs to ask a question about it, they come to you, which is happening. But the one thing about coronaviruses at the time is that it was largely understudied. And so there was a need for the type of expertise that I felt like I could give. And it also would mean that I would be able to work outside of the pressure vacuum that comes from, the HIV field, for example, or influenza even. And so that's why I chose the field. I really chose the field because there was this niche for understanding basic viral immunology to propel vaccine development forward. And interestingly enough, when I started, the MERS outbreak was actually happening. And so although it was not to the level which COVID-19 outbreak is, there was still this kind of trickling steady state need for understanding for coronaviruses. So I thought that the field just really fit perfectly with my interest, expertise, and the kind of, purpose that I wanted to have in science.
So this MERS outbreak that was happening in 2014 when I started at the Vaccine Research Center was actually the second of a similar type of outbreak for coronaviruses. The first of which some of you may remember was the SARS outbreak that tallied over 8,000 cases worldwide. And then the MERS outbreak that started in 2013 that has since tallied over 2,000 cases worldwide. So history continued to repeat itself. And then we had this large family of viruses in this coronavirus family that basically were just waiting to emerge into humans. And actually many groups, including Ralph Barrett's group at the University of North Carolina, had described that this had the potential to happen, especially with viruses in the SARS-like subfamily of coronaviruses. And so I like to think of the 2019 emergence of SARS-CoV-2 that has since turned into a global pandemic, somewhat like the virus's way of saying, I told you so. The good thing, though, is that vaccines exist. And the only way that something like this could ever end is, like I said in this video, with most or all of us being immune.
A global race to find treatments for the sick and a vaccine to protect the healthy.
This ends with most or all of us being immune to this virus, and ideally that's through a vaccine.
Can solutions that usually take years
we are trying to move as fast as we possibly can
be found in a matter of months?
It is an extremely revolutionary approach to get into a phase 1 clinical trial 66 days after the sequence of a virus is released is a world record. And the reason why we had to go about it in kind of this record speed is because when you think about the numbers, in order for most or all of us to be immune, or about 70% of the population to be immune, that means that about 40 million people around the globe would have to succumb to COVID-19 if we were allowed that immunity to happen via natural infection, which is simply unacceptable. And so one of the things that we like to remind people is that it isn't just about yourself when you think about vaccination, but also for the benefit of your community, whereby vaccines can reduce transmission and therefore make it safer to resume our normal activities. And the way that we do this taps into my kind of nature of community service, in that vaccines and getting vaccinated is community service. It is community service because once you're vaccinated, you become one of the green people on this slide. You become a part of an immune population whereby the blue people have the ability to get infected. And if a large population of the blue people who are not immune have the virus circulating in their population, then they could potentially get infected. And then unfortunately, because of the mortality rate or the morbidity rate of COVID-19, it is about 1% of people that will have to die. However, when you become one of those green people by getting a vaccine, there's a chance that in your community, there's lower transmission of the virus, where essentially the green people become roadblocks and the virus can no longer spread throughout that community. And so you can see how on a very community specific and then country specific and then obviously global, the global and worldwide way that this type of population or herd immunity can start to really decrease the level of pandemic spread of this particular virus. And so we went about our approach to making a vaccine for SARS-CoV-2, actually before SARS-CoV-2 was ever even a thing. And we do that by the the prototype pathogen approach to pandemic preparedness, whereby you do research on a particular virus within a given viral family. You make generalizable knowledge to that particular virus, and then you're able to apply that to all of the viruses in that viral family. And that way you can really speed the development of a vaccine shall a pandemic occur. Dr. Graham and I summarized this in the Journal of Clinical Investigation, and I like to call it the plug and play method, where we haven't got to the point where we could vaccinate people before a pandemic, at least for coronaviruses, but we are at least to the point where we were able to plug and play what we knew about MERS into the in the onset of this particular pandemic. So there were three things that we knew that we wanted for a vaccine that we could be prepared in case of a pandemic. And those three things were, firstly, a vaccine that was fast. So that is a technology that must uphold some level of being able to be produced in vast quantities fairly quickly, and then reliable. A technology that has some level of reliable manufacturability standard, and then universal, in the italicized way of how we think about a universal vaccine, in that maybe it's not preemptive, but at least, and can provide protection against future outbreaks, but at least it does have this kind of universal approach, or it's a universal solution in that what we understand about vaccine development prior can be transplanted in this instance with this new virus. And so the first reports of a respiratory virus outbreak happened at the latter parts of 2019. And then the sequences of that virus, which were then called the 2019 N or novel coronavirus, were published in January 10th. And we were able to go into a phase 1 clinical trial in only 66 days. And so this part of this slide and this timeline is what is the media view of what happened during this vaccine development process. However, the real view happened for the 6 years or 7 years prior to the pandemic in all, in which extensive work on MERS coronaviruses and other coronaviruses was done so that we were able to move very quickly in this moment. So let's talk just a little bit about that work. So on the left side of your screen is a coronavirus that has a spike protein on the surface. And then on the right side of your screen is a cell, like a cell that's in your body, that has receptors. And so they receive the virus via their receptors. And so the spike protein actually acts like a key to the lock. So it unlocks your cell and allows for the virus to get inside of the cell and then replicate. And that's how people become sick with COVID-19. However, if you want to block that interaction in any way, you can actually prime your immune system to see the spike protein without having your body get sick at all. And that it would be by way of vaccination. So we actually had no idea what a high definition structure of a spike protein looked like. So we could look at cartoons and we could see, very slightly by looking at a virus under a microscope, but into like an atomic level detail, there were no spike structures that were published in the literature when I got to the laboratory in 2014. And then we were able to publish the first coronavirus spike protein structure in 2016, I believe, which was the spike structure for a human coronavirus that is one that kind of causes the mild symptoms or mild flu-like symptoms every single flu season. And then the literature kind of lit up with various laboratories from all over the world publishing different spike structures, and it really increased our knowledge on how we might want to make a really good vaccine for coronaviruses, particularly by targeting that spike protein. And so at the same time of which George Gao's group, which is the director of the CDC in China, published his coronavirus structures, spike structures for the MERS and the SARS coronaviruses, which were again, the two that had jumped from their animal reservoirs into humans and caused these regional outbreaks. And were so important for our understanding around how to make a vaccine against viruses that might cause that type of regional outbreak. At the same time, we published our spike structures for MERS, but remembering not just that we were publishing the structures, but as the title says, we were also defining the immunogenicity. So that is, how does the spike make immune responses? And also characterizing the spike by way of using not just the spike as it appears in nature, but we designed a spike protein that would be a really, really good vaccine antigen, or the thing that you give to someone when you give them a vaccine. And so the way that we did this is by a way of structure-guided stabilization of pre-fusion MERS coronavirus spikes. And so we took the sequence of the spike protein, as you can see it here as it's laid out linearly, and we compared it from what we knew about the HKU1 spike. So that's the first spike that we ever published, and MERS and SARS, which are very similar, at least in a particular region. This region down here in the yellow and orange, that sits pretty adjacent to this really cool peptide called the fusion peptide. And we knew that where that region is this spring. So you can imagine that when this protein does its dirty work or inserts itself into the human cell, part of what happens is it does that by releasing this blue fusion peptide by springing up this particular mechanism here. But if you were to add various mutations in that area, you can prevent that from happening. And that is the type of protein that you want to present to the body by way of a vaccine, because that is exactly the functionally relevant protein that your body sees when it sees a virus, and you want to be able to prime your immune system to the best protein possible. And so how do you know that it's the best? Well, you insert certain mutations. We actually stumbled across these two prolines, or you will hear it being called 2P mutations, that when they were inserted into the spike, they actually prevented the spike from flipping, as you can see here, into its more elongated form. So now you have these beautiful mushroom-shaped proteins that are ready to prime the immune system. And I don't talk about the work around the immune system with MERS, only to say that they look very good when you gave them to mice, and they look better than what nature could provide by what is on top of a virus. And so with that, we took those two proline mutations and we put them into the backbone of all of the other spike proteins, two other ones, including HKU1 and SARS. And then also, importantly, the one that was considered to be potentially emerging as defined by Ralph Barrick's group, which was in the SARS-like COVID coronavirus family. So we had the protein. We knew that it made really good immune responses. But if you're in a pandemic, remember, you want to be able to go fast. And you want to have a technology that's reliable, at least from a manufacturability standpoint, and something that's italicized universal in that you can plug and play. And giving protein vaccines actually is neither of those. Because every single time you want to make a protein in large quantities that is good enough to give to a human, like for example, with the Novavax vaccine candidate, it takes some time to get to that point. And so we actually wanted to use the human body as the vaccine protein factory. So we took our spike protein with those two proline mutations in, and then we purified the mRNA, and then we encapsulated that mRNA in the lipid nanoparticle using Moderna's proprietary formula. And if you give that messenger RNA to a person, and the person's cells reads it, then they make the spike protein, and then obviously their immune system kicks up and makes this really, really good immune response to that spike protein. An immune response that is so good that hopefully it will protect you against COVID-19 or the viruses that cause COVID-19. So we were able to decide on the sequence for that vaccine in only 3 days after the sequences were published, largely again because we already knew what to do. We knew to drop those two proline mutations into the backbone of that sequence. And that was the way that we'd done it before. And we were almost certainly convinced that we would be able to do it in a similar fashion with this new virus. We then also wanted to, again, solve the structure of that spike protein to confirm, like, yes, this is homogeneous, pre-fusion stabilized spike in the same way that we had before. The interesting thing, though, taking it back from just the vaccine, which you are hearing about every day on the news probably, and also right now in this talk, is that this spike protein with those two proline mutations led to us isolating the first in human monoclonal antibody therapy that is being given and developed by Eli Lilly. We were able to also use that spike protein to make serology diagnostic tools, and on top of it being in the backbone of the the Moderna vaccine, it's also in the backbone of the Pfizer and BioNTech vaccine, the Novavax vaccine, Johnson & Johnson, Sanofi, and several other vaccines around the globe. So the utility of a protein that is very highly characterized and also is pre-fusion stabilized with certain mutations is so good when you think about vaccine development. And so after deciding on that sequence, we were also able to go in the direction of confirming that the vaccine elicited immunogenicity or immune responses in mice. And I don't talk about any of that data, only to jump to the non-human primate, mostly because the data that we got from the non-human primates is starting to be reconfirmed as more and more data are coming out with humans, where when we give the vaccine at 100 micrograms, which is exactly the dose that you get if you go get the Moderna vaccine, comparing that to a much lower dose at 10 micrograms per monkey, and then comparing that to a group of animals that did not get any vaccine at all, then you give a lot of virus into the nose and also into the lung. And you beg the question, how much viral replication is happening both in the lung and also in the nose? On the left-hand side, you can see that by day 4, after the vaccine, those animals were given that large amount of virus into their nose, the vaccinated animals had completely cleared the viral replication from their lungs. And it wasn't just for the lungs, but also by day 2, the animals that got the 100 micrograms or the clinically relevant dose had also cleared it from their nose. And so this was the first time in the literature that it was shown that limiting viral replication in both the lower airway and the upper airway in the nose and potentially have implications for disease and also for transmission. And the only reason why I bring that up is because we're currently debating, so to speak, although it's not really a debate, it's just that on a day-to-day basis, more and more data come out, and more data are basically confirming what we saw in the monkeys about a year ago, whereby the vaccines are preventing asymptomatic infection or transmission. And these kind of studies in humans over time are the reasons why over time we are also able to, as vaccinated people, remove our mask, because it's very clear that we are not having virus replicate in our nose and therefore are not transmitting virus onto people if we come in contact with it. And so Moderna was also at the same time had initiated the production of the clinical grade material that would then be shipped in only 41 days after the production started. So this goes back to the utility of that platform being fast and reliable in the case of a pandemic. And the phase 1 clinical trial starts. And I normally don't show the phase 1 clinical trial data because we're in phase 3 right now and we have an efficacy signal that's 94%. But I do like to show these data, especially in instances like this, because I like to describe kind of in the stepwise fashion how all of these data are constantly showing us exactly what is happening, and also have led us to the point where we were so confident in the ability of this vaccine to be efficacious in people. And so in this small cohort of, I think it ended up being about 100 people in a phase 1 clinical trial, we gave 100 micrograms in people that were 18 to 55 years old, and that is highlighted here, and showed that they have really high level antibody responses. These types of high level antibody responses, although I don't show that bar on the graph, are far exceeding what you see in people who just got infected naturally with COVID-19. And it wasn't just for people that were 18 to 55 years old, but also for people that were over the age of 56. And you can see that after 2 doses of the vaccine, again, even older adults have those very high levels of antibody responses. And that's not just at the peak. So this particular time point here is at the peak, or the 2-week time point where the CDC says fully vaccinated. But also after that, as you continue to bleed those people over time, you can see that there isn't really a significant dip in the antibody responses over time through to the month 6, which is published so far, and you'll see like the year data coming out soon. Nevertheless, so we're eliciting very potent neutralizing antibody responses that are not just high level, but also long lasting. And so in the phase 3 efficacy trial, which started, I believe, last July, so at the end of last June or last July, so we're about a year from that phase 3 efficacy trial starting, we were able to vaccinate 15,000 people and then compare them to people that got a placebo saltwater injection. And those volunteers were then going about their normal everyday lives. And if they got any indication that they might have COVID-19, they came back to their clinical trial physician and they were diagnosed with COVID-19 or not. And so the vaccine efficacy is calculated as the percent of COVID cases in the placebo group as compared to the vaccine group. In this particular example, where there are 2 volunteers that got COVID-19 in the vaccine group and 18 in the placebo group, it's a 90% efficacy. In the case of mRNA 1273 or the Moderna COVID-19 vaccine, it was a 94.1% efficacy, because as you can see, the placebo group over time, cases continue to accrue throughout that phase 3 interim analysis. Whereas on oppositely, in the orange with the mRNA 1273, there were only 11 cases total in this interim analysis, which measured out to be a 94.1% efficacy rate. And when we also look at adverse events, which is a hot topic also, because people are very concerned about the safety of these vaccines that have been developed, quote unquote, so rapidly, there are things that do happen. We don't call them adverse events, we just call them events, because they are actually the types of things that you would expect as your body is ramping up an immune response to the spike protein and to the vaccine, where you might get pain or swelling or redness at the injection site, or you also might get, after your second dose in particular, more systemic events where you have fever or fatigue like I had, or muscle ache, and etc. And so in conclusion, following the promising phase 3 efficacy results, mRNA 1273 is currently being used in multiple countries. And although I do like to use the Bill Nye GIF, it is very clear, especially more and more data continue to come out and more questions start to arise. That's the beautiful thing about science, is that your job is actually never done. So we're currently assessing multiple different things. And the reason why I have this immune correlates of protection highlighted is because I don't have time to talk about those data. But if for people who are really interested in the nitty gritty of immunoassay assessment with the serum from non-human primates and also from humans, you should check out that paper. And without further ado, I will thank everyone on this team here who has essentially rocked it out with me for the last 6.5 years. Dr. Barnett Graham, who has been a tremendously amazing mentor to me. And my fellowships throughout my postdoc, which, as I said on that first slide, it kind of always goes back to the resources that that help to kind of fuel your career forward in this way. And not only that, but also mentors who are able to, literally and figuratively step to the side and allow you to be kind of in the center and kind of lead your own destiny. The VRC has somewhat miraculously, in my opinion, started an entire VRC-wide COVID-19 response. And everyone in this slide has their hand in some portion of our response, whether it be antibody therapeutics or the mRNA vaccine candidates, other vaccine candidates, even communications. They're even swapped with that. And so we have a large network in the Vaccine Research Center, and all of these talented people are to thank for all that has been happening throughout this entire time. And then lastly, our collaborators and contributors, firstly, Moderna, who manufactures and distributes and develops the vaccine, but then all of our academic collaborators, whether it be our structural collaborators at UT Austin or Scripps, UNC that do our animal or mouse challenge studies, or even internally at the NIAID, where our animal pathologists are, and people who do our electron microscopy, and et cetera, who are everyone in this network who we were collaborating with before or even started to collaborate recently has helped along with this project. And of course, a special thank you to all of the clinical trial volunteers. And I actually usually have a line here, although I think I deleted it here, but also thank you to everyone who has gotten vaccinated, whether it be with the Moderna vaccine, the Pfizer, or otherwise, because you are becoming one of those green people that are becoming a roadblock for this virus and helping us to, in your own way, get out of this pandemic. And as was said in the introduction, since we're talking about trajectory, I am now heading off in less than a month, scary, but to Harvard Chan School of Public Health, where I will be assistant professor in immunology and also take on a professorship at the Radcliffe Institute of Advanced Study, an associate membership at the Reagan Institute, where I will continue to do work just like this, actually continuing to finish off some of this work, but also broaden my horizons a little bit to study coronaviruses from a universal perspective and other viruses as well. And I don't think that we have time for questions, but I just want to say thank you so much for attending this today, and thank you for hosting me.
Wow. You know, Dr. Corbett, I am just so inspired by and thrilled for, excited for you. And I love that chart because I know what we're going to see from 2021. It's just going to continue to be this upward trajectory. There's no limit to what you can do. You know, what I heard today is, you know, the simple conclusion could be, well, you were at the right place at the right time. But your story shows that it's much more than that. Your story shows at least not just your 6 years of research, but the volume of individuals who had incredible dedication, who really picked the right areas to study. I love your example of how you picked something without the fanfare. You wanted to build that niche that really could explore a novel, unanswered question. And now the world is on it. But you're ahead of the pack. And I just know with your inquisitiveness, with your abilities, you're just going to be picking those right spots in the future. And we're all the beneficiaries. So it's in that spirit that I put on my gloves here to handle a very, very heavy weight. I want to present to you the 2021 Chancellor's Innovation Award, which I could not think of a more deserving person in the world. And we're so grateful for you spending time with us today, sharing your story. So I'd ask everybody to give the virtual applause for you. And we're just so appreciative of your innovation and how you've been a role model for so many. Thank you for saving so many lives and having such great positive impact and really focusing on the inequities, helping bridge that gap. We look forward to continuing with our collaboration with you. And I now hand it over to Vice Chancellor Borger for closing this out. Patricia?
Thank you so much. This was just a wonderful experience. So thank you, Chancellor Mone. Thank you, Dr. Corbett. We truly look forward to welcoming you back to Milwaukee or to Milwaukee for lunch sometime soon when we can all do that. So I thank everyone for attending this virtual awards ceremony today. And I look forward to seeing you all in person the next time we do this. Thanks to Dr. Corbett's work, we'll be able to see each other in person again safely. Thank you so much. Have a wonderful afternoon and evening. Take care, everyone.
Thank you.